Hello and welcome GCSE students to another online lesson, this time focusing on the topic of microscopy. Alright, so if you are already familiar with me and how these videos work, please feel free to skip this portion of the video. However, if you are new here, my face looks new and these video lessons are new to you, pause the video and have a read of these 4 points that will explain to you exactly how best it is to use this video. As soon as you finish reading it, then press play again and we'll get straight into the lesson. Alright, so a quick look at what we're going to be doing in today's lesson. So the first thing we're going to look at is we're going to look at some of the parts of a microscope. We're then going to work out how to convert between different types of units. So we're then going to look at a couple of key definitions before moving on to some microscopy calculations, and then finishing with our fast fire 4 or our summary questions. Alright, let's get into the video. Question number 1. So on screen is a diagram of a microscope with features labelled A through E. Your task is to label those features on the microscope. Now there are 2 options for you, a red and a blue task as always. The red task is for you to copy, trace or print the diagram below and label it using the labels in orange. And then the blue task is for you just to write out a list of your answers, so A equals B equals and so forth, using those hints in orange. As always, pause the video, have a go at completing it, and then press play when you are ready for some answers. Alright, hopefully now you have done it and you are ready for some answers, so let's have a look at what they were. A was the eyepiece lens, B was the objective lens, C was the focus adjuster, D was the stage, and E was the light. And just a side note as well, in order to calculate the total magnification, it's important to remember you must multiply the value of the eyepiece lens by the objective lens. Let's get on to question number 2. And here we are. Now this is a very tricky and slightly longer task than we are used to, but it's going to be something that's going to be really important anyway. So in this activity we're looking at conversions between different types of units. So in the same way that we have centimeters, meters, kilometers, we've got units that get smaller as well. So your task is to use the orange boxes at the bottom of the screen to fill out the table in front of you. The red option of the task is to copy out the table and fill it in, and the blue option is to present the information in your own way. So whatever you want to do, you can quickly just jot out the information or you can present it as some sort of written piece if you so wish. I do not mind, but either way, pause the video and have a go at completing this activity. But bear in mind, this is a little bit of a tricky activity, so if you do get stuck, don't worry about watching it through and seeing some answers. So pause the video and have a go. Alright, now here are the answers, so very well done if you managed to get them. Please do note as well that these are purple, which means you should be self-assessing your work. I forgot to mention that for question number 1, so if you haven't self-assessed your work for question number 1, make sure you go back and do so, but with a separate colour pen. Let's go through it. So taking each one row at a time. So the first one was given to you 1/1000th of a meter. In standard form, this is 10 to the minus 3. That means you move the decimal point from 0 3 spaces. The unit is millimeter, the symbol is mm, and you can see something this small with the human eye. Underneath that we have 1/1000th of a meter, which is 10 to the minus 6, and this is known as a micrometer, and the symbol is that funny U with the M following it, and you can view this with the light microscope. And then lastly we have 1/1000th of a meter, which is 10 to the minus 9, and that's known as a nanometer or an nm, and you need an electron microscope to view this. And please note, to get from nanometer to micrometer, you have to multiply by 1000. To get from micrometer to millimeter, you have to multiply by 1000 as well. And if you're going down the table, you divide it by 1000. And that's going to be really important when we're doing unit conversions. Alright, so question 2 was a long activity. Question 3 should be nice and short. So all you have to do here is match the key term to its definitions, and there's a question down at the bottom of the screen for you to answer as well. So it should be pretty straightforward. The red and the blue tasks are the exact same here. Copy out the definitions and match them to their keywords. Pause the video and off you go. This one shouldn't take too long. Alright, hopefully you managed to get that. So just for you to check your work, The magnification is the number of times larger the image is than the real object, and the resolution is the ability to distinguish between two separate points. And a side note as well, both the magnification and the resolution are better in electron microscopes, and that means we can see really small organelles like ribosomes on electron microscopes. The light microscopes you probably have in school, you can only see the cell and maybe the nucleus as well if it's a really large cell, but you will need electron microscopes to see any of the organelles which are significantly smaller than that, such as ribosomes. All right, on to question number 4. So on question number 4, we have 4 questions, and each of them rely on the I am triangle. So that I am triangle, hopefully you've been through it in school, but it's image stands for the I, actual size stands for the A, and M stands for the magnification. Now I assume you know how to use it. If not, quickly listen to what I'm going to say. So in order to calculate the image, we put our thumb over the I and we do actual size times magnification. In order to calculate the actual size, we put our thumb over the A and we can see it's image divided by magnification. And in order to calculate the magnification, we put our thumb over the M and it's I divided by A, which is image size divided by actual size. So using that formula triangle, I want you to have a go at completing questions number 1, 2, 3, and 4. Please be careful with number 4 as well, because it's asking you to switch between units as well. So pause the video here, grab a calculator if you need to, and answer these questions with full workings, because that's both your red and blue task. Off you go. All right, assuming now you have the answers. All right, now let's go through some answers. Remember, these are in purple as well, so grab a separate color pen and make sure you are checking them as you're going. Let's go through them together. So for question number 1, we have to calculate the magnification. So in order to calculate magnification, we do image size divided by actual size, which means we do 15 divided by 10, which gives us an answer of times 1.5. For question number 2, we are again calculating the magnification. So we do image size divided by actual size, which means we do 5 divided by 0.4 to give us an answer of 12.5. And then on number 3, we are looking at actual size. So we do image size divided by magnification this time. So that's 3 divided by 500 to give us an answer of 0.006 millimeters. And then for question number 4, we were calculating the actual size. So image size divided by magnification, which is 20 millimeters divided by 1,000. But remember, in this case, the answer was asking for the units to be in micrometers. So we then had to times it by 1,000 to finish with our answer of 20 micrometers. Now, very, very well done if you got any of those questions right. Question number 4 especially is going to be very much a higher tier question, because it's a 2-stage calculation, because then we had to change units as well. But very well done. Students always find these topics a little bit tricky, but awesome job if you manage to get it done. And now lastly, we have our final 4 questions, which is always used to assess our understanding of the topic. So I don't need to read them to you. Pause the video and have a go at answering those 4 questions. And when you are ready again, press play. All right, hopefully you've done it now. Grab a separate color pen. Let's go through some answers. So when looking for ribosomes, what type of microscope would you use? You would use an electron microscope. How do you calculate the total magnification of a light microscope? It's the eyepiece lens times the objective lens. What is 0.00025 nanometers in millimeters? Remember, we times it by 1,000, then times it by 1,000 again to give us an answer of 250 millimeters. And then how do you calculate the image size from the actual size and magnification? What you do is you do actual size times magnification. A very, very, very well done, students. If you got this far in the lesson, I appreciate it's not always easy, especially with some numbers, and especially some of those standard forms getting a little bit tricky, 10 to the minus 3, 10 to the minus 6, 10 to the minus 9. If you do have any questions, please do ask your teacher or give us a join on our WhatsApp channel, and we will be able to support you further there. But very well done for making it this far in the video. Until the next video, take care.